Welcome back. This is the 14th part of the digestion absorption. Welcome to Laksha YouTube channel. Okay, let us start with the protein digestion. In the previous part, we learned how proteolytic enzymes are activated, which are present in the pancreatic juice. Pancreatic juice consists of proteolytic enzymes like trypsinogen, chymotrypsinogen, and procarboxypeptidase. How they are activated to from inactive form to active form, how they have become violent. Okay, let us now see how proteins are digested. Proteins and protease and peptones, which are the protease and peptones are partly hydrolyzed proteins. You have already learned in the digestion of proteins in the stomach. Protease are formed. Protease and peptones are formed when pepsin acts on the proteins. Okay, these protease and peptones are the partly hydrolyzed proteins which are present in the chyme. The proteins, proteases, and peptones which are present in the chyme, now they reach the duodenum. Okay, pyloric sphincter relaxes. Now the chyme, small amount, every time small amount of chyme it comes to the intestine, and here and these proteins and protease and peptone proteolytic enzymes of pancreatic juice and intestinal juice act. What is proteolytic? Lysis means breakdown, proteo means protein digesting enzyme, which are present in pancreatic juice and intestinal juice. Now they act on them. They act on proteins, protease, and peptones. Let us see those equations, how these enzymes. Okay, these are the enzymes of trypsin, chymotrypsinogen, carboxypeptide. These are present in pancreatic juice. Amino peptidase, tripeptidase, dipeptidase. Okay, these are present in intestinal juice. Okay, now what happens here? The proteins and protease and peptones are converted to tripeptides and dipeptides by trypsin, chymotrypsin, carboxypeptidase, and amino peptidase. Okay, where are these trypsin, chymotrypsin, and carboxypeptide? Okay, they are present in the pancreas. These are the pancreatic enzyme. Now the tripeptides are converted to dipeptides and amino acids. Okay, where are tripeptides? Tripeptidase is present. Tripeptidase is present in intestinal juice, which converts tripeptides into dipeptides plus amino acids. Now the dipeptides are converted to amino acids by enzyme called dipeptidase, which is also present in intestinal juice, and finally amino acids are formed. Amino acids are the end products of protein digestion. What are the end products of protein digestion? Amino acids. Okay, next slide. Okay, amino acids, the end products of digestion of proteins, namely amino acids, are formed in the small intestine. Okay, where amino acids are not formed in our stomach, amino acids are formed in the small intestine. Then the digestion of fats. How fats are digested? Okay, bile salts of bile help in emulsification of fats. Okay, earlier we learned bile composition. Bile has bile salts like taurocolate and glycocolate. Okay, those bile salts of bile. Okay, those bile salts of bile, I will show you here. Bile salts of bile acting on fats. Observe this one. Observe this slide. This is the fat, and these are the bile salts. They are acting on them. As a result of that, emulsified fats are formed. Okay, bile salts of bile help in emulsification of fat. That means, what is meant by emulsification? The breakdown of fats into very small micelles. Micelles are formed. Okay, bile also activates lipases. Okay, which lipase bile activity? The 2 marks one, very important. These 2 points together, 2 marks question. Okay, bile also activates lipase. Bile has no enzymes. It is very important. Even though bile juice does not have enzyme, it is very important for fat digestion. How? Okay, bile salts of bile help in emulsification of fat, that is breakdown of fats into very. Why should the fat molecule, larger fat molecules must be converted to very small micelles? Because to increase the surface area. Okay, now as the fat molecule is broken down into smaller one, now the surface area increases. Therefore, enzymes, lipases can act on them easily. Okay, bile also activates lipase of pancreatic juice and intestinal lipase of pancreatic juice and intestinal juice. The lipase of pancreatic juice is called pancreatic lipase, which is also called steaoption, and intestinal lipase. These lipases act on emulsified fats. Observe this one. Okay, these are the emulsified fats. Now lipase acting on the finally what are formed? Fatty acids and glycerol are formed. These are the end products of fat digestion. Observe here. On the emulsified fats, lipase act and diglycerides are formed. These diglycerides are converted to monoglycerides by the action of lipases. Now the monoglycerides are finally converted to fatty acids and glycerol by the action of lipase. The end products of fat digestion are fatty acids and glycerol. What are the end products of protein digestion? Amino acids. What are the end products of fat digestion? Fatty acids and glycerol. Okay, that's the end of fat digestion. Finally, these are the digested fats are absorbed into the epithelial cell layer. We will discuss about that in the next one. Okay, now come to the digestion of carbohydrates. Carbohydrates in the chyme are hydrolyzed by pancreatic amylase into disaccharides. Carbohydrates which are present in the chyme are hydrolyzed by pancreatic amylase into disaccharide. What do you mean pancreatic amylase? The amylase, the carbohydrate digesting enzyme present in the pancreatic juice is pancreatic amylase. Earlier you learnt one amylase. Okay, I think you can recall that. Okay, the saliva has one lipase that is called salivary amylase. One amylase that is called salivary amylase. Okay, now this is a pancreatic amylase. This amylase is present in pancreatic juice. Now it acts on the carbohydrates present in the chyme. And now the intestinal disaccharidases. What are the disaccharidases? I will show you here. Observe this slide. Okay, these intestinal lipases are also called. Okay, these are also called. Okay, brush border enzyme. These are also called brush border enzymes. What are they? Sucrase, lactase, maltase. These are called brush border enzymes. Observe this one. The intestinal disaccharidases means. Okay, I told you one code called sumala, sucrase, maltase, lactase. These are called disaccharidases. They act on disaccharides. What are the disaccharides? Sucrose, maltose, lactose, and convert them into monosaccharides. Okay, we will see how in this equation. Look at this one. Carbohydrates are converted to maltose, sucrose, lactose, which are the disaccharides. Don't confuse disaccharides with disaccharidases. Disaccharidases are the enzyme. Okay, these are the carbohydrate disaccharides. Okay, which are made up of two components. Okay, carbohydrates by this amylase is not salivary. This amylase is pancreatic amylase. Okay, pancreatic amylase converts carbohydrates into maltose, sucrose, and lactose. Okay, now the maltose is converted to by the enzyme called maltase, which is one of the brush border enzyme or disaccharidase. I will show you this one. Okay, observe here. This is the maltase enzyme. Maltase enzyme which is present in the brush border of the enterocyte, which is the cell which is present in the gut is called enterocyte, and convert maltose into glucose plus. Okay, glucose and glucose, two glucose molecules are formed. Observe here, glucose and glucose are formed when maltase acts on maltose. Now come to the next one. Sucrose. It is commonly called as cane sugar. When you take sugarcane juice, okay, that sugarcane juice consists of sucrose. That sucrose is converted to glucose plus fructose. Okay, there are two units are present. That's why it is called disaccharide. Maltose has two units called glucose and glucose. That's why it is called disaccharide. These are the monosaccharide. Okay, now the sucrase acts on sucrose. Sucrase is also called invertage. It was given in previous NEET exam. Okay, now it is converted to glucose and fructose. Let's look at the next one. Lactose, which is commonly called milk sugar. Milk protein is the casein. It is a milk sugar. Okay, the lactose, which is a milk sugar, is converted to glucose and galactose by the enzyme called lactase. These three enzymes are called disaccharidases or brush border enzymes which convert. These are the disaccharides into monosaccharide. The end products of carbohydrate digestion are glucose, fructose, galactose. What are the end products of protein digestion? Amino acids. What are the end products of fat digestion? Fatty acids and glycerol. What are the end products of carbohydrate digestion? Glucose, fructose, galactose. Now we move on to the next one. Digestion of nucleic acids. Okay, nucleic acids, I think what are the nucleic acids? DNA and RNA are called nucleic acids. and they are also digested. To digest the nucleic acid, there are enzymes called nucleases. Where are they present? What are they? Okay, DNAase and RNAase are called nucleases, which are present in the pancreatic juice. They act on nucleic acids, which are DNA and RNA, and form the nucleotides and nucleosides. Look at this equation. Nucleic acids, okay, DNA and RNA, okay, which are acted upon by nucleases, DNAs and RNA, which are present in pancreatic juice and converted to nucleotides. The nucleotide is composed of, you might have learnt in botany when you are studying DNA molecule, structure of DNA molecule. Okay, nucleotide consists of nitrogen base, phosphate, and sugar. Okay, three compounds. Nitrogen nucleotides by the enzyme called nucleotidase, which is present in very important one, which is present in intestinal juice. Now the nucleotide is converted, tides are converted to nucleoside plus phosphate. Phosphate was removed. Now it has only two components. Now the nucleoside is acted upon by nucleosidase. Nucleosidase is also present in pancreatic juice like nucleotidase. And now what are found? Finally, sugars and bases are formed. What are the sugars here? Okay, in our DNA molecule there is a sugar. In RNA molecule there is a sugar. The sugar in the DNA is called deoxyribose. The sugar in the RNA is called ribose. And what are the nitrogen bases? You are familiar with the nitrogen bases: adenine, guanine, thymine, cytosine, uracil. Okay, these are called nitrogen bases. And finally, in this way, nucleic acids are digested by the action of enzymes present in pancreatic juice and this one. Okay, now, okay, let us see finally, okay, what is going on here. Okay, protein digestion. We started with protein digestion. How proteins are digested by the enzymes of pancreatic juice and intestinal juice. Okay, here the proteins are digested to first converted to tripeptides and dipeptides. Okay, proteins, protease, and peptones are converted to tripeptides and dipeptides by the action of the pancreatic enzyme. Then tripeptides are converted to dipeptides and amino acids by the action of tripeptidase. Okay, tripeptidase is present in intestinal juice. Next, dipeptidase is also present in intestinal. In this way, finally, proteins are digested to amino acids. Then after that, okay, fats. Okay, fats first by the action of emulsification. Okay, fats are converted to emulsified fats by the bile salts. The emulsified fats are converted to diglycerides by lipase. Now the diglycerides are converted to monoglycerides by lipase. The monoglycerides are converted to fatty acids and glycerols also by lipase. Finally, the fatty acids and glycerol are formed at the end of fat digestion. Next one, digestion of carbohydrate. The carbohydrates are converted to maltose, sucrose, lactose by pancreatic amylin. Now the disaccharides called maltose, sucrose, lactose are converted to monosaccharides by disaccharidases. Okay, next one is the digestion of nucleic acid. The nucleic acids like DNA, RNA are converted to nucleotides by nucleases. Then okay, nucleotide is converted to nucleoside plus phosphate by nucleotide, and nucleosides are converted to sugars and bases by nucleoside. Okay, that's the end of digestion in small intestine. Okay, if you look at here, observe the end products. Proteins are converted to amino acids finally, and starch is polysaccharide is converted to monosaccharide as glucose, and fatty acids, fats are converted to fatty acids and glycerol. These are the end products of digestion of bio-macromolecule. Finally, the bio-macromolecules are converted to simple diffusible forms, absorbable form. That is the purpose of digestion. The purpose of digestion to convert the complex molecules present in the food into simpler form. With this digestion is completed. The digestion is completed in the small intestine, and no digestion takes place in the large intestine. Okay, that's the end of this part. Okay, like this video, share it, and subscribe to others, and subscribe to Lakshya, share to others, and subscribe to Lakshya YouTube channel. Okay, keep watching. Okay, until then, take care of yourself.